Welcome everyone to this week's edition of the Extracellular Vesicle Club. This week is a treat for me in part because I was just on a call earlier today and somebody was saying, you know what, we don't know a whole lot about EVs in pregnancy. And I said, well, this is a great advertisement for today's EV Club because we're actually going to be talking about EVs and specifically here in gestational diabetes. And so I'm very happy to introduce our presenters today. So Carlos Salomon is going to be presenting the work, and he's joined by his colleague, Laura James-Allan. So Carlos is at the University of Queensland in Brisbane, Australia, and Laura then is at UCLA. And we're really looking forward to hearing about this recently published work and its implications for the community. So Carlos, I'd like to hand this over to you. And I'd just like to remind everyone to put your comments and questions in the chat box. And then that way, at the end of the presentation, we'll be able to let you unmute, let you show your video and interact with our scientists here today and learn more from them. So Carlos, please go ahead.
Thank you, Ken. And well, first of all, thank you for the invitation, and thank you for all the time that you invest in the EVClub. I think it's a fantastic initiative that help a lot of our community, right? So which establish discussions and also maintain us up with the literature, which is always something that we have to do. And also because Laura is here, thank you also, Laura, for joining this presentation. That is fantastic. I'm presenting mostly all of your work. So is there any tricky question at the end of the presentation? Laura is the person who will probably answer this question. So as Ken mentioned, this particular study, the title is A Novel Technique Using Chronic Infusion of a Small Extracellular Vesicle from Gestational Diabetes mellitus cause glucose intolerance in pregnant mice. This is a great collaboration, mostly within three different institutions. So the University of Queensland, of course, where I'm based, University of Alba in Chile, and the University of Colorado with a group of Thomas Johnson and Teresa Powell group, and of course Laura, who actually I had the pleasure to host Laura for, I think, 6 to 7 months, a couple of years here in Brisbane, and did mostly of the experimental work of this study as well. Cool. Yeah. So this is the study. So this is essentially the first page of the published study. So you can see that was published last year in November in Clinical Science. And I mentioned this because one of our electron microscopy images was also the cover of that particular issue of the Clinical Science, which is fantastic for us. I think good for the promotion of our community, and hopefully next time we're going to have a cryo-EM picture image rather to a TEM. But I think that was fantastic, and we have been followed the citation, but also the number of access in the website. So we know that it has been a good impact in the fields as well. So I would like to start with this because I'm not really sure somebody knows Helen Keller, but Helen was a disability rights advocate. And this probably is not new. This is something that a lot of people have heard as well about. Alone, we can do so little. Together, we can do so much. And I think this is a good reflection of this particular study. Because as I mentioned, this is a wonderful collaboration between three different institutions, all over the world, in which we essentially collect the sample, isolate VVs, characterize VVs, everything that we did here in Australia. Then the majority of the functional assays were performed in Colorado, the US, and the immunoassay in Chile, in the University of Alba. So this is, hopefully there are a lot of ECR and MCRs join this particular club. And this is something that hopefully you're going to learn as well, how to interact and collaborate with different people, and taking expertise from different labs around the world and all push forward to a specific goal. So this is something that is a very great collaboration that we have, and hopefully we're going to have more studies to share with you in the future, right? So I'll give you a little bit of context. So this is essentially us at the University of Queensland. So we're based in the UQ Center for Clinical Research, and we are a group that we focus in extracellular vesicle research. We have, in the last 2 weeks, rebrand our lab. So now we are the Translational Extracellular Vesicle in Obstetric and Gynecology group, which reflects essentially our vision for the next 5 to 10 years as a group. And what we do essentially is focus in extracellular vesicle in translational. So how we can implement our bench discoveries in clinical practice. And for that, what we do is isolate different type of extracellular vesicle from, as you all know, from different bioelectrical fluid, from cell condition media. For this particular presentation, we do a lot of placenta. We culture syncytial trophoblast cells, and also placenta explant, in which we isolate EVs from the cell condition media. And in terms of the bioelectrical fluid, we isolate EVs from plasma from pregnant women at different gestational age as well. Then, of course, we characterize the type of EVs that are present in our preparation, and this is no surprise. We know that extracellular vesicle is an umbrella term that involves different type of extracellular vesicle and with a high heterogeneity as well. We are also a biomarker discovery lab, so we focus a lot on the molecules that carry by this extracellular vesicle. We have a mass spectrometry program running, trying to identify protein associated with this extracellular vesicle, and also a genomic program focused in nucleic acids, mostly in microRNAs through small RNA sequencing. And in the last 2 years, we have been also focused a lot on therapeutics, trying to use EVs as a therapeutic agent, but also EVs as a carrier of different therapeutic molecules as well. So this is essentially our overview of the lab, which we are based here. So this is in Brisbane, Australia, and we are more than happy to collaborate as well in case that you want to interact with us. So keep in mind that the extracellular vesicle, and this is probably not new for the audience, but this is something I always trying to point it out, why is a hot topic, and has been a hot topic for the last 10 years because of the capacity to interact and involve in cell-to-cell communication. So always I mention this is kind of an envelope, and then you have inside information that you can deliver to different, in this case, of the extracellular vesicle, different tissues and organs, right? So as I mentioned, we focus a lot in pregnancy. So obstetrics is one of our arms of our program. And of course, we are interested in characterize extracellular vesicle, circulating extracellular vesicle during gestation. And if you're talking about pregnancy, probably one of the main things that you need to focus on is what is the role of the placenta during gestation, right? If you're not familiar with the human placenta, so I introduce you. I have been working with placenta for a long time. I did my MPhil in placenta, my PhD in placenta, my first postdoc in placenta. So I really think this is a remarkable organ. If you're not familiar, so essentially the human placenta have two sides. The fetal side, in which you have faced the fetus essentially, and then is connected to the placenta through the umbilical cord. And all the placenta is a fetal tissue. And then you have a maternal side, which is in direct contact with the maternal circulation. And then you have this giant cell, this syncytial trophoblast, which is in direct contact with the maternal circulation. So what we are very interested in all the molecules, including the extracellular vesicle, that are in direct contact with the maternal circulation that are released from the human placenta, right? And I'm gonna go through a summary of the different aspects of the extracellular vesicle role during pregnancy later on. But this is something that we're very interested in. We have been also focusing gestational diabetes because even though is the higher incidence, is the highest incidence in terms of complication of pregnancy during pregnancy, which over 10% of all the pregnant women develop gestational diabetes and is associated with maternal obesity and overweight. The mechanistic is not really understood. We know that there are different kind of molecules released from the placenta that can cause actually glucose intolerance and change insulin sensitivity during gestation. But of course, we don't know the whole picture, right? So we are very interested in determining what are the main mechanisms associated with changes in insulin sensitivity during gestation, right? So in that, what we know is, for example, pregnancy hormones and other placenta factors can increase glucose intolerance during gestation, but also change insulin sensitivity during gestation. However, I would like to point out that all the studies in which they try to correlate the level of placental hormones and the degree of glucose intolerance or insulin sensitivity during gestation show that they do not correlate at all, suggesting that perhaps there are other mechanisms and other factors that can be associated with changes in insulin sensitivity during gestation. And now it's have been almost 10 years in which we have been essentially proposing this idea that the extracellular vesicles have a role in this phenomenon, right? So this is a summary of a lot of work that we have done in the last 10 years. And I was just going to give you a brief summary of what we have done. So we know that, for example, placental cells are very sensitive to changes in the extracellular environment. So one of our initial work, we isolate placental MSCs and also syncytial trophoblast cells from placenta. We expose them to different oxygen level, and we know that low oxygen tension can increase the release and change the content of the extracellular vesicle, mostly small EVs like exosomes. And these EVs can interact with target cells, interacting with endothelial cells, skeletal muscle cells, and vascular muscle cells, changing the release of cytokines from, for example, endothelial cells, change the migration and the response to insulin in skeletal muscle cells as well. So after that, we have also profiling the level of circulating extracellular vesicle in different complications of pregnancy, and we identify that gestational diabetes, preeclampsia, fetal restriction, and also preterm birth is associated with a specific signature of extracellular vesicle, which potentially can be associated with pregnancy outcomes as well. The other thing that we're very interested in, of course, we are able to characterize a level of placental EVs that are present in maternal circulation. But also we know that in particular in gestational diabetes, you can see that not only you have placental EVs, you have also EVs coming from other sources. And one of the main contributing factor of these extracellular vesicle levels during pregnancy is extracellular vesicle coming from the adipose tissue. This is something that you have to keep in mind when you work with EVs from biological fluid, because essentially you are isolating EVs coming from different origin. So when we isolate EVs from pregnant women, we have a proportion of this EV that actually coming from the placenta, but also we have EVs coming from other sources as well, right? So what we have been working on for a while now is the hypothesis that we think that there are the placental EVs that are released from the human placenta very early during gestation that can interact with insulin target organs and regulate insulin sensitivity during gestation. That is probably the main concept that we have been working on, right? So to give you a little bit of evidence of that particular hypothesis, I'm gonna very briefly introduce these two studies. So the first one was an in vitro study in which we provide evidence that the placental EVs can induce changes in insulin sensitivity in vitro, and then this study in which we also provide evidence that EVs can induce glucose intolerance in vivo as well. So in this particular study that was published a couple of years ago, 4 years ago actually, 5 years ago, what we did is isolate EVs from normal and GDM pregnancy, and we have two main cell types as a target cells. So we have a skeletal muscle from subject with normal insulin sensitivity, and we have a skeletal muscle from patient with diabetes type 2. And then what we did, something very simple. So we incubate EVs on the cells, and then we determine the glucose uptake in response to insulin on this cell type. So here you can see the normal skeletal muscle in white, the diabetes type 2 skeletal muscle in black, right? So this is just the characterization of our model. You can see that, of course, normal skeletal muscle respond better to glucose uptake in the presence of insulin compared to the patient with diabetes type 2, right? So this is just confirming the insulin sensitivity model in our cell type, right? And then when we incubate this with EVs from normal pregnancy, you can see that you can see much different in normal in skeletal muscles from a patient with normal insulin sensitivity, but you can see a decrease in the patient with diabetes type 2, right? Which is very interesting because we know that also normal pregnancies is associated. So it's a degree of glucose intolerance and insulin resistance in normal pregnancy, right? And that is completely normal, especially the first 20 weeks of gestation. So you can see that when you incubate these skeletal muscle cells with EVs from GDM pregnancy, you abolish essentially this glucose uptake, stimulate glucose uptake in the presence of insulin. So provide some evidence that actually these circulating EVs in GDM pregnancy can cause insulin, change in insulin sensitivity, at least in vitro. But of course, we know that not always when we do stuff in vitro also works in vivo, right? So, and that is one of the reasons that we start to collaborate with Thomas and Teresa several years ago to try to determine if these changes that we see in vitro actually can be reflected in vivo. So the idea is to inject or infuse extracellular vesicle in vivo to determine that can induce changes in glucose homeostasis and glucose intolerance in an in vivo situation as well, right? And this is essentially the second study. So we published this 2 years ago, and then you can see that I'm gonna go through the new study just in the next slide, but you can see how these EVs that were infused in non-pregnant mice can induce insulin glucose intolerance from GDM pregnancy, EVs from GDM compared to the normal pregnancy, right? Provide not only now in vitro, but also in vivo evidence of that, right? So, and that essentially was the rationale for us to perform this particular study. So we know we have in vitro evidence that circulating EVs in GDM pregnancy can induce change in insulin sensitivity at the level of skeletal muscle cell. And we know that when we inject or we infuse these EVs from GDM pregnancy in an in vivo situation, you can see also changes in level of glucose in these mice as well, right? However, the in vivo experiment that we performed was done in non-pregnant mice, and we know that essentially because the human placenta, so the placenta is there in a pregnancy situation, that essentially physiology is quite different. So the idea of this particular study was determine if you can infuse extracellular vesicle from normal and GDM pregnancy in a pregnant mice, you can actually mimic this insulin sensitivity or change it in insulin sensitivity state, right? So here what we did, we isolate EVs from normal and women with gestational diabetes. Samples were collected at the moment of the diagnosis, around 20 weeks of gestation. So to avoid any co-funded with different treatment for gestational diabetes. So all the samples that were collected were before any type of treatment for these women. Then we isolate the EVs. And then I'm gonna talk a little bit more about this mini osmotic pump, how we infuse EVs to determine if they can induce changes in glucose intolerance in these mice. After that, we also determine the effect of these EVs on the secretion of insulin from the beta cell of the pancreas, and also insulin signaling in skeletal muscle as well. Of course, because we are infusing human EVs in a pregnant mice, one of the big questions is, well, essentially any kind of change that you can observe in insulin, in glucose homeostasis, is due that you are inducing some pro-inflammatory state in this mice. So we did some basic immune response experiment, just to trying to determine actually that is happening or not, right? So this is essentially graphical after of our study, and this is the stuff that we're going to go through in the next 10 minutes, hopefully. So, so you can see this is essentially the clinical characteristic of the patient involved in this study. So you can see that all of these women that were diagnosed with GDM, you can see a higher levels of glucose when we perform the oral glucose tolerance test, which is essentially the criteria of the diagnosis for gestational diabetes, right? So, and then you can see that we have the same number of patients from normal and GDM pregnancy. So what we did here, it's important to highlight that we isolate EVs for every single patient. So this, you can see 24 independent isolations for each of the group. But then because we're gonna test this in an in vivo situation, we pull all the sample and then we infuse the same amount of EV in each of the mice. This is something that you have to consider as well. So in terms of the characterization, so you can see that we isolate EVs using a gradient. And the reason to perform a gradient that people actually are no, it's not that common anymore because it's time consuming, because usually the yield is quite low because of course what you're doing is enrich a specific population of extracellular. Something happened, Ken? No?
Oh, no, I'm just...
Okay.
no worries. Everything's good.
Okay, cool. So, the reason that we use a gradient to isolate EV is because even though you decrease the yield, seems to be you have a more pure population of extracellular vesicle, and because we're gonna pull all the EV as normal or GDN group, we were not actually worried about the amount of EV that we can get, right? So that is the reason that we use this particular method that I'm happy to discuss after the presentation. So, and then we confirm essentially that there are EV from the placenta present in our preparation. You can see we use a placenta alkaline phosphatase, which is a specific marker for the placenta. So all the EVs that will reach the maternal circulation from the human placenta will be positive for blood. So highlighting that we have that in our preparations. And also our EVs were positive for CNI and THG101, which are two proteins associated with extracellular vesicle. And then we did some morphological analysis using electron microscopy just to determine that we are working with the EVs. After that, we continue with our characterization. And for that, we did some exo-view analysis to determine it's a single EV analysis, which what the exo-view does essentially is determine the level of different tetraspanin, different EVs that are positive for protein associated with extracellular vesicle. In this case, the kid come with CD81, CD63, and CD9. So this is the representation of the analysis. You essentially have like a chip in which you have in the bottom of the chip, you have these three proteins associated with EVs. And then usually the chip coming with also a secondary antibody, which is a fluorescent antibody, against the same protein. So what we did is replace one of these antibody, so the secondary, the CD63, for a PLAP antibody. So for us, to trying to address the question is what are the number of placental EVs that are present in our preparations, right? So actually the exo-view also not only give you a very beautiful images of your EVs, but also give you very useful data. So you can see that the level of T63 PLAP were higher in gestational diabetes compared to normal pregnancy. And then the different population of EVs defined by the protein that are positive mostly in the surface of the EVs, you can see also were in general higher in GDN compared to normal pregnancy. This is something that is consistent with our previous publication and also have been confirmed with other groups around the world. Also, the exo-view give you information about the size. So you can see that we obtain vesicles, small EVs, around 60 nanometers in size, which is something very interesting because When we did the same analysis by a nanoparticle tracking analysis, our size was around 100 nanometers. And we know that the NTA usually overestimate a little bit the size of the EVs. But I guess we all agree that we are working with small EVs in this particular preparation. So after that, we infuse. This is something important, and this is something of the novelty of the study. We have a great discussion about, okay, we're gonna test these EVs, and we're gonna check the effect of EVs in this in vivo situation. And for that, how are we gonna infuse or inject EVs in this animal model, right? So it's a nice supplemental table in the supplemental material of the paper, in which we did a little bit of literature review of a different technique and how much EVs have been injected, mostly in a vivo situation. And of course, when you inject EVs, you have to also decide how much EVs you're going to inject, how many times you're going to do it, right? And for how long. So which is something that probably doesn't mimic really a in vivo situation. So we decided to use this approach, which essentially is use a continuous infusion of extracellular vesicle using a mini-osmotic pump that is in the jugular vein of these mice. And we infuse EVs for 4 days. So we use the same concentration of EVs from normal and GDM pregnancy. This is something also is up to discussion after the presentation, because we observe higher levels in gestational diabetes, right? But just to avoid that co-funding, what we did was use the same concentration of normal and same concentration of GDM, right? And after 4 days of chronic infusion of this extracellular vesicle, we did a glucose tolerance test, and you can see that mice that were infused with excess of EVs from GDM pregnancy, you have 30 minutes and 60 minutes, the glucose level are higher compared to normal pregnancy and our control, which was PBS, right? Here you have the raw numbers of the graphic there. So provide more evidence that these EVs from GDM, the circulating EVs from GDM, can induce glucose intolerance in this in vivo situation under this approach, right? So then what we did was quantify the level of insulin in these mice, and also the level of plasma adiponectin. So adiponectin is a protein that is released mostly from the adipose tissue and have insulin sensitivity effect. So low levels of adiponectin have been associated with higher insulin resistance, and we know that the low levels of adiponectin also have been observed in women, in overweight and obese women as well. So we thought that insulin and adiponectin is essentially the two minimal analysis that we have to perform in these mice. It's very interesting. So we didn't observe anything in the plasma adiponectin, but you can see the plasma insulin. A high level of plasma insulin in mice that were infused with EVs from normal pregnancy compared to the GDM pregnancy, which is quite interesting for us, right? So we know that insulin is secreted by the beta cell in the pancreas. So maybe it's something that something is doing the EVs from GDM interacting with this beta cell of the pancreas that for some reason this beta cell cannot increase the level or cannot increase the secretion of insulin, right? So we explored this a little bit further, and this is something that was developed by Fred at the University of Colorado, which is I don't remember how many years of experience working with beta cells, but many years. And you can see actually that the changes in insulin, in plasma level of insulin, is due that is a lack of secretion of insulin from the beta cell of the pancreas in the beta cells isolated from the mice that were infused with GDM EVs. So you can see here that we used the two different concentrations of glucose, 2.8 and 16.7 of this beta cell of the pancreas. And then you can see that the beta cell that were isolated from mice infused with normal EVs, you can see an increase in the secretion of insulin, and this is completely abolished in the presence of the EVs from GDM, right? Which is quite interesting for us. Then we determine if these changes in glucose concentration and glucose intolerance is associated with changes in insulin signaling in the skeletal muscle and the adipose tissue. So this is in the supplemental material. We didn't observe any difference in the adipose tissue, but you can see that the phosphorylation of AKT in the muscle is different between the normal versus the GDM pregnancy, which potentially can explain the difference in the changes in the insulin sensitivity that we observe in these mice. Of course, as I mentioned, we are infusing human EVs in a mice model, so that means that potentially we could we could cause some immune response in these mice. So to trying to elucidate that, what we did was isolate T cell from the spleen from these mice. And then what we did was determine if actually the EVs are inducing T cell activation or immunosuppression using concavalin A, or if we can see any difference in terms of the T cell proliferation. So the graph is just the representation of the flow cytometry analysis here. So you can see how we didn't see any effect when unstimulate and then we stimulate the T cell with concavalin A. You can see that we cannot see any immunosuppression effect from normal EVs or GDM EVs. And similar results were also observed in the T cell proliferation. That provides some evidence, at least, that the changes that we observe in glucose intolerance, in the level of glucose, and changes in insulin sensitivity is not because we are inducing some kind of immune response from these mice. At least in part, give you some idea of this, right? Then, of course, we did also we check some characteristic of the placental and fetal characteristics of the mice that were infused with the EVs, and we didn't see much difference in the fetal weight or no difference in the fetal weight, placental weight, and also the ratio, right? Suggesting that perhaps because the insulin changing insulin sensitivity was not strong enough to affect actually the fetal characteristics of the mice involved in this study, right? Just to summarize, what we have is GDM is associated with a higher concentration of small extracellular vesicle and is linked to maternal glucose intolerance. However, the mechanism by which small EVs regulate maternal glucose homeostasis in pregnancies is unknown. Using a novel approach involving continuous intravenous infusion of a small extracellular vesicle with a mini-osmotic pump, we demonstrate that small EVs isolated from GDM women cause glucose intolerance in pregnant mice because of impaired ability to stimulate compensatory insulin secretion. In part, we think that is happening. And also our findings suggest that circulating small EVs contribute to the attenuated insulin secretion and more pronounced glucose intolerance, which underlie the pathologies of the gestational diabetes. Just to finalize, I would like to just mention that what we are proposing is that during early gestation, the human placenta have the capacity to sense this microenvironment, and in response to that microenvironment, will encapsulate specific packaging of molecules in this extracellular vesicle. This extracellular will reach the circulation and interact with insulin target organ, including also the beta cell in the pancreas, change the insulin secretion, change how also the skeletal muscle, insulin signal in the skeletal muscle, and end up with a change in insulin sensitivity during in women that will develop gestational diabetes later on. So we think that is kind of part of the puzzle that we're trying to elucidate here. Finally, I would like to thank to my group. As I mentioned, we have rebranded our lab. So if you are interested, just go to the website and then you're gonna find more information about what we do and the different areas of our program that we are developing in the UQCCR. Finally, just to thank the different collaboration and different funding bodies that allows us to do all these experiments. Especially, I would like to thank to Samya, who have been, driving this, for the last 5 years. She did the PhD with me in gestational diabetes. Now she's doing a postdoc as well. Also Dominic and Andrew, who have been fundamental to the progress of our program as well, and essentially everyone that's involved in our lab. Thank you.
Well, thanks very much, Carlos, for this presentation, and to you and Laura and all of your collaborators for the excellent work. I think you're addressing a very important problem here, and we've all learned a lot from your work. And I see there are a couple of questions that have come into me directly. As I mentioned a little bit earlier, we had a bit of an incident. So I would just like to remind everyone, if you do have a question for Carlos and Laura, please put it in the chat box. And I might just go ahead and read these, but feel free to follow up in the chat box, and I might experiment with unmuting in a few minutes here, but I don't want to have somebody screaming at us. So Carlos, let me just start with a question about the implications here. So what implications do your findings have for glucose intolerance during pregnancy? Do you think that it might be a matter of infusions from healthy blood that might help, or is it going to be more a matter of blocking these it's the bad effects of the bad EVs?
Yeah, thank you for the question. This is something, probably I have more than one answer for that, right? I think there are two aspects here. One is the knowledge aspect, right? So we don't fully understand how changes in insulin sensitivity during pregnancy occur. So as I mentioned in the presentation, we know that there are placental hormones, there are different pro-inflammatory factors that contribute to this change in insulin sensitivity and changes in the glucose tolerance of these women. But we don't have the complete picture. So I think from the knowledge point of view, it's important that we discover and we still elucidating that seems to be that the extracellular vesicle released not only from the placenta but potentially from the adipose tissue are actually part of this phenomenon, right? So the question is, can we do something with the EVs if we know that they are participating in this overcompensating insulin resistance during early gestation that potentially will end up with a woman being diagnosed with gestational diabetes? One option is, as you mentioned, is to identify the mechanism. For example, we can identify that they are the placental EVs that are encapsulated with a specific microRNA, a specific protein, or something that can induce this. Can we actually block the interaction of these placental EVs with the target cells? Can we block the specific packaging of this molecule that potentially can be delivered to the target cells? So, and transform maybe this diabetic EV phenotype in a normal EV phenotype, right? And the other is, can we treat these women with normal EVs, for example, to trying to avoid the development of gestational diabetes? This is something that I think is up to discussion and I'm still exploring. The other thing is about not only trying to avoid the changes, this drastic changes in insulin resistance during gestation, not during pregnancy, because if you address that, you're also gonna avoid different complications in the future. What we know is women that actually are born from a pregnancy with gestational diabetes have a 5 or 6-fold changes increase to also develop gestational diabetes during their pregnancy. And if they're carrying a girl, that girl also have a higher chance to develop gestational diabetes and develop complications of different metabolic complications later on. And the cycle essentially continues. So if we have a window, especially during early gestation, that we can avoid that, I think it will be fantastic.
So we have a question here, and I have had some people write me directly. So let me just allow unmuting here. We'll do a little experiment. Okay, so I have a question here from Ravin Fisher. Do you want to go ahead and ask Carlos your question? I may have scared people off from from interacting here, but anyway, the question is about the cargo. So, have you been able to define the cargo here in these EV populations? Do you, and maybe I'll expand on this question a little bit. Do you think that this is a cargo effect, as in something that is being delivered to recipient cells, or is it rather some sort of a surface interaction where you're stimulating signaling pathways? What do you think there?
Potentially can be a little bit of both. This is something that we have been also thinking for a while, how we can answer that particular question. So what we observed in the previous study, which we use non-pregnant mice, that when we isolate the skeletal muscle from these mice, you can see a clear change in the microRNA profile in the mice that were infused with GDM EVs compared to the normal EVs, suggesting that potentially is some kind of delivery of microRNA that are different in GDM compared to normal, that can induce the change in insulin signaling that we observe in the skeletal muscle from these mice. So we have been working on this for a while, and we have been also publishing several papers on the cargo of the EVs. And I have to tell you that, and probably you know this better than myself, when you work with cells, so we isolate cells from placenta, for example, right? And then you have GDM placenta, normal placenta. You can see a defined signature associated with GDM compared to the normal. When we're trying to explore and expand this in circulating EV from plasma or serum from these pregnant women, the variation from different women from the same group is just too high to define the signature, which has been one of the issues that we have. Even though I still think that is the delivery of the cargo, but I cannot discard that potentially some kind of signaling in these EVs coming from the placenta, most likely, that can be also interacting with the receptor of the surface of the target cell that can be interacting. This is quite interesting. And I guess the only thing that we can do is trying to answer one question at a time. So identify and focus in one molecule that we think that potentially can be involved in this phenomena, overexpress this molecule, knock out this molecule, and determine if we can lose the effect on changing insulin sensitivity.
Fascinating. Yeah. So that in that case, then you might be able to block a microRNA or enhance a microRNA depending on its up or down regulation. Very good.
Yeah. The other thing is quite interesting for us as well is, and you have seen that in other publications, is when if you back to the culturing of placental cells, right? So trying to avoid, because when you isolate EVs from plasma or serum, you have EVs from everywhere, right? If you just isolate EVs from placental cells, and then whatever is happening in the cells, what we think that is reflected in the EVs. So then the idea for us was, okay, so whatever protein or microRNA that are changed in GDM in the cells, perhaps that is reflected in the EVs, and that particular microRNA is probably driving the effect, right? What we observe that in general, you can see a reflection of change in the cells is reflected in the EVs. But you can see also a clear specific packaging of microRNA in the extracellular vesicle, which is quite interesting as well for us. And we have been trying to develop that story a little bit further as well.
And of these microRNAs that you are seeing, whether they're differentially regulated or whether you're seeing, this specific packaging, are any of these microRNAs part of the placenta specific cluster, or are they other microRNAs entirely?
Yeah, so I can tell you some mix, because so we focus in the microRNA part of the C19MC cluster, which for us will be actually awesome if we can see a clear signature. But what we found is a mix of microRNAs. Specifically, one of the part of the C19MC cluster are part of this signature, but there are others as well. Yeah, it's quite interesting. Maybe I'm not trying to ask a question to you, but especially when you work with microRNA and also with proteins or whatever cargo that you want to elucidate the role of that particular molecule inside of the EVs and then how they interact with the target cell, you need to identify a molecule, not only that is different between normal and GDM in my case, but I guess need to be in enough abundance to do something. And this is how probably you have to elucidate what will be the molecule that you have to focus on.
Yeah, exactly. Because you could have something that would work very well as a biomarker, but maybe it's not doing much of anything. And then it would be a wild goose chase to go after that one. Yeah, for sure.
Absolutely.
I have a, you know, I know the C19MC cluster you just mentioned, that are the members the same in mouse?
No, it's different. I think in mouse is the C14MC. That is specific for
it's a different chromosome. So this would be maybe just in primates, or I think I used to know that and I've forgotten.
Well, you know, I probably have heard maybe 20 different presentations for Joel Sadowski in Pittsburgh, who also probably one of the experts on that particular cluster, but also I kind of forget.
Yeah, fascinating though. I mean, because there are so many microRNAs that are pretty much ubiquitous, but then you have these few, you know, that are just expressed in the liver, or just in red blood cells, or just in placenta. It's really fascinating. Yeah. We have another question here, and this is a fascinating question for me. And it's a little bit about cargo, but maybe about cargo that's on the outside of the EVs. And so two of the really hot topics that I've seen in the past couple of years, and we see this reflected, I think, in ISEV annual meeting submissions as well, are the topics of lipoprotein EV associations, and then also the so-called EV corona. So the things that, you know, this could be lipoproteins, it could be other things as well that are sort of on the outside of EVs, associate very loosely. So if I understood correctly, you were using density gradient centrifugation to purify your EVs, which is really one of the most rigorous ways that you can do it. And yet there's no perfect method. There are always going to be some things that co-purify. What do we know? What do you know? Because I don't know anything, but what do you know about how lipoproteins are changing in GDM? Is there anything that's known about those profiles? Like, are HDL the same, LDL, other lipoprotein particles?
Yeah, look, that's a fantastic question, and I don't want to, you know, I feel that every time that I ask a question, I try to answer yes too many things. But definitely, this is something that we should consider. We know that, for example, HDL is the one that carry microRNAs, I think. So this is something that we have to consider. We also know that there are no perfect methods to isolate EVs. And we know that even a density gradient, you can see in the fraction that you are collected, some of the lipoprotein involved. Some I don't want to say contamination, because essentially at the end of the day, it's biology. So it's part of what we do. Probably you have the same issue when you work with virus, I guess. But this is something that we have to consider. The other thing that we have to consider is, of course, level of LDL or HDL is highly dependent of the BMI of these women as well, right? So, and we know that overweight and obesity is associated with higher incidence of gestational diabetes. So this is something that we need to check. What I can tell you is BMI in normal and GDM for the group of women that we work here was no different in the group, right? But I don't have more information about the lipoprotein profile on this particular set.
Yeah, very interesting. And you're right. I think you make a very good point there, that we maybe should not be talking about contaminants, but rather, you know, co-travelers, friends. You know, maybe these EVs are exerting their effects through the associations they have as well. So a lot to explore there too.
Absolutely, yeah. I'm surprised no one have asked about the immune response that we did.
Aha. Okay. So ask the question that you were expecting, and then
no, look, I think this is something that also, especially because now, you check the clinical trial website, it's a lot of EVs using in clinical trials as therapeutics, right? So the question that always people ask is how immunogenic are these EVs? And my reply is, which EVs you are talking with? If you're talking with EVs from immune cells, I'm sure they are immunogenic, right? you're talking of EV from other sources, probably you're not going to have a much immune reaction, I guess, right? So I think one of the interesting things about this particular experiment that we perform is, keep in mind that we isolate EVs from plasma. So that means this EV is coming from everywhere. We have a lot of EVs from adipose tissue, from the placenta, from endothelial cell, maybe some from platelets, and maybe some proportion from immune cells, right? So what I think that we didn't see any effect, which probably was a little bit of a surprise, is because even though we have EVs from immune cells in our preparation, that proportion is not enough, or maybe it's not different between normal and GDM to cause some kind of immune reaction, at least in this in vitro situation.
Now, how long were you infusing again?
4 days.
4 days, yeah. So what do you think would happen if you were to infuse one day and then again 2 weeks later and then again? Well, I mean, we're talking about mice here, I guess. But if do you think that repeated exposure might lead to some responses that you would not see after 4 days?
So you're suggesting that this kind of immune adaptation in the second day, that is the reason that we don't see anything, right?
Or you know, what if you do come up with an infusion therapy for, you know, for human pregnancy, and it would be given repeatedly, you know, would there be development of a response? When
I don't know.
Not see
maybe
that just a few days. So that's I think that's a, you know, that it just depends on exactly how you would implement the therapy, whether that would be something that you would have to consider. But the
other thing, the other thing is, it's have been also several studies in the published in the literature about injecting human EVs in mice. So this is not the first one. Probably the first one in what we infuse.
Yes.
The other thing is also, you know, I got this comment all the time that there's a lot of information about the immunoregulatory effect of EVs. And, you know, Clotilde work a lot with dendritic cells EVs, right? But again, this is EVs coming from an immune cell. So of course, I guess you have to consider that when, I think you cannot, especially with the EVs, you need to take in consideration what is the source and the origin of the EVs to determine, well, these EVs, and maybe you can add isolating with this approach, you can see this particular effect, right? Also, how wide can be applied? I don't know, actually.
And actually, we get a question right now from Arpita Burke, who asks, well, it says the EVs injected in the mice were isolated from plasma, containing, of course, EVs from a lot of different sources. So do you think injecting placenta-specific EVs would have an effect? Different effect.
I think that, well, that is our next, I think. So what we did, there are two studies. So the first one that we published in FAST, we used non-pregnant women as a control. So then we assume that the pregnant versus non-pregnant is the effect derived for the placental EVs, right? Which of course have a pro and cons with that approach. But what we like to do is isolate placental EVs and determine if that actually placentally is driving this particular effect. This is something that we have been discussing a lot in our group, and hopefully we're going to be able to have some data to support this hypothesis in the future.
That's great. Yeah, and Arpita has really anticipated this, so that's so wonderful. So I think that this is bringing us towards the end of our hour here, and I would just like to invite Laura, you, and also Carlos. So maybe I'll start with Laura. Where do you see this headed next? I mean, we've just heard about another direction that this is going in, a way that we can refine this, understand a little bit more about the sources. So Laura, what do you see as the next steps here in understanding how these EVs are influencing the glucose intolerance?
Yeah, I mean, I think Carlos has spoken quite a lot about, you know, the next steps, but I think the obvious one is to look at the placental specific EVs. There are some difficulties with that because at that gestation that we're looking at, there aren't that many within that population of EVs that we isolate. I also think, obviously, looking at, you know, the cargo and trying to focus on that would be really interesting. but also the source, you know, like we said, the placental specific EVs, but also seeing potentially, is there a difference in the maternal source of EVs? So maybe from the adipose tissue, or maybe other sources that we haven't really considered yet as well.
Wonderful, wonderful. And Carlos?
Yeah, you know, I don't want to repeat what I said, but I feel that in vivo value distribution is one of the missing data that we need to address as well. So not only the effect, but how we know that, for example, the EVs are interacting with the insulin target tissues. I know that we are seeing the final effect, but I feel now probably we have more tools to identify a better in vivo value distribution of different EVs that we're infusing or injecting in this mice. I think it would be fascinating to run a study in vivo value distribution of EVs using different approach to infuse or inject in mice. So when you infuse EVs versus you inject in the tail of the mice, when they reach the systemic circulation, is the same value distribution or not, for example. I think this is something that will be very interesting to know.
Fascinating. Well, thank you both. Laura, thank you. And thank you, Carlos. And please pass along our congratulations to the whole team for this work that, again, I think is very important and is taking us closer to the clinic where EVs can be used in a very good way. And thanks to all who joined. Please know that we have a YouTube channel where this video will be archived. You can ask more questions there if you'd like to. And otherwise, please join us again at a future EVClub. So thank you all. Hope you all have a good rest of the week and good luck with your research. Take care now.
Thank you. Thank you, Ken. Bye.